Thank you for the kind introduction, and thank you to the audience for attending this talk. So in the next 30 or so minutes, I'll try to introduce you to the GenScript capabilities and solutions for therapeutic antibody discovery and development. Oops. So the agenda is basically, I'll give you a little bit introduction to GenScript, who we are, and then more about the GenScript solutions for antibody drug development, and then how you may partner with GenScript on your developments. So GenScript was founded in 2002 in New Jersey, US. We have
Yeah, it doesn't always work.
Okay, yeah. We have multiple locations around the world. We have 2 manufacturing locations in US. We have one on the East Coast, one on the West Coast. We have 2 manufacturing locations in China. Our research use services and also GMP capabilities, a lot of them are built there. We also have manufacturing location in Singapore to serve our Asian Pacific customers. And we're a relatively old company. We have been around for 20 years. We have very strong experience in antibody development. That was one of the first services GenScript launched. We have done over 150,000 projects. We have completed also 15 clinical stage projects, and we have done 14 therapeutics antibody licenses. This is a little bit more about GenScript, and again, I'm going to focus in this presentation, I'm going to focus only on our CRO and CDMO capabilities and our product capabilities. We do have other entities inside the company. So as I mentioned, GenScript was founded in 2002. In 2013, we launched our Bestzyme company, which is focused on synthetic biology, mostly focused around human food and animal food supplies. Then in 2014, we officially launched Legend Biotech, cell therapy company, which was previously housed within GenScript. We became public in 2015 in the Hong Kong Stock Exchange. In 2017, Legend Biotech signed an agreement for co-development with Janssen for cell therapy. We launched in 2018 our ProBio CDMO division. In 2020, Legend Biotech went public on NASDAQ. In 2021, we got more investment for our CDMO division and Legend Biotech. And in 2022, Legend launched their Carvykti cell therapy together with Janssen. So I just want to mention this to give you an idea who GenScript is. But again, I'm going to focus mostly on our services and products related to research and clinical developments. So if we look at general antibody drug discovery workflow, typically would go through something like target identification or screen many targets, then validate them, which ones are druggable and which ones are not. Once you select your targets, you may decide to develop some antibodies for these targets, basically antibody generation, a lot of screening, antibody engineering, a lot more screening. Eventually, stable cell line would be developed. Then animal preclinical studies, IND filings, and then if everything goes well, you would go to clinical trials and clinical development. So GenScript as a company, with our services and product, we can support you through this whole development, starting from target identification all the way to GMP production, clinical trial support in GMP production. So these are some of the services, basically a map. I'm not going to read all of these, but basically we have a lot of services and products around target identification. Typically you would do something like gene level analysis, protein level analysis, cell-based assays at this stage. We can support all of those. We have lead generation and optimization services and products, basically preparing the plasmid, the antigens, the antibody development, screening, optimization, optimizing the properties, and so on, then preclinical studies and clinical studies. For target identification and validation, we have a number of offerings. Here we have a lot of services around PCR, of course, PCR and qPCR for fundamental techniques for gene analysis. Then we have some support for NGS sequencing, CRISPR knockout libraries, and so on. For protein analysis, we have variety of services and products, and then for cell-based assays, we have a lot of cell line development services as well, as we have over 300 off-the-shelf cell lines that you can just purchase. And again, for NGS development, we have capability to create libraries, enrichment libraries, and basically we can create hybridization-based libraries or amplicon-based libraries. For knockout screens, we can create CRISPR gRNA libraries, and we can deliver them to you either in form of plasmid or a virus. All this is powered by our proprietary semiconductor technology. Essentially, we have a semiconductor chip that can produce over 8 million oligos in parallel. So we can produce those oligos in parallel. We can use them to create various libraries, either for NGS or for gRNA and so on. For protein level analysis, we have full suite of gel and Western blotting products. This is, as you know, Western blotting is fundamental technique. Very often you would use it early stage to see if your target is expressed, maybe do some semi-quantitation. We have excellent gels for electrophoresis. We have all the related products around them. We have devices for transferring the proteins from the gel to the membrane, basically doing the blotting for automated staining. We have a device for automated processing of the membrane. So full suite of products around Western blotting. Of course, Western blotting requires a lot of antibodies, so we have very strong antibody development capabilities, anything around polyclonal and monoclonal antibodies, primary and secondary antibodies. We have technology for rabbit monoclonal. We have excellent anti-VHH antibodies for anybody working with VHHs as a drug, and so on and so on. We have also conjugation and labeling services. Very often you may need proteins either for your interaction assays or for antigens and other techniques. So we have various expression systems. We have eco-expression system. We have done over 50,000 projects already producing with eco, various enzymes, antigens, cytokines, and other proteins. We have excellent insect development expression system. We have expressed proteins as high as 300 kilodaltons. And we have excellent proprietary HEC and CHO expression systems. And with some of these systems, we can do very fast expression. We can basically, if you give us a sequence, we can deliver to you a protein or antibody within 5 days. We can produce it within 5 days and ship it to you. So for target identification, of course, very often you may do a lot of ELISA screens. So we can produce a lot of antibodies for ELISA. We can also produce a lot of ELISA assays. We can do sandwich, direct, indirect, competitive ELISAs, and so on. During COVID times, we actually launched excellent neutralization assay for COVID, and so on. We have very strong capabilities in that area. Cell-based assays, as I mentioned, we have very strong CRISPR knockout capabilities, knocking capabilities. We can do deletions. We can do basically constitutive expression, inducible expression cell lines. And as I mentioned, we have over 300 off-the-shelf cell lines, basically a lot of them around immune checkpoint, GPCR cell lines, FC receptors, and so on. And those are available for delivery within a week, essentially. For lead generation optimization, we also have strong offerings. These are some of the services and products related to that. As I mentioned, plasmid design and construction, antigen preparation, antibody development, optimization of the antibodies with various mutation libraries, and so on. We are one of the leaders in gene synthesis. We have a variety of offerings in the gene synthesis area. We can produce pretty much any gene. For very wide genes, we also have the gene brick service, which is basically breaking the gene into fragments to make it faster and easier to produce. And then we have excellent assembly kit, basically cloning kit, gene builder that can allow you to assemble those fragments in the appropriate sequence into the plasmid. In addition to that, we can do a lot of plasmid preparations. We can produce any plasmid in any quantity, essentially, with various quality levels all the way to GMP. We can produce your vectors. We have a lot of products around mutagenesis. We also have excellent ORF cDNA collection. So if you need any kind of cDNA, you can look at our ORF collection. Most likely is available there. We recently launched an excellent instrument for plasmid preparation. So in case you prefer to do the plasmid preparations, purifications yourself, instead of outsourcing that, you can use this instrument. It's one of the few instruments on the market capable of doing something like that, automating completely the plasmid purification. Basically, you only need to have your E. coli lysate. You put your E. coli lysate inside the tube of this instrument. We have cartridges containing all the reagents that you need. You put the cartridge in, you push a button. 2 hours later, you have up to 24 samples purified. So the system is modular. You can have up to 4 modules connected to one controller, and basically each module can purify up to 6 samples at a time. So 4 modules can give you 24 samples, and basically you can do that within 2 hours. Plasmid levels are up to 1.5 milligrams of plasmid can be purified, anything from 300 micrograms to 1.5 milligrams. Runtime is less than 2 hours, and the toxin levels are very low, and supercoat ratio is usually very high. So this in case you would like to have automation in-house. And again, we are best known as a services company, but we have very extensive portfolio of some of them very innovative products and very interesting systems for automation. This is one of them. For producing antibodies, of course, we have very strong capabilities. We can do anything from regular hybridoma. We can use single B-cell capture to produce rabbit monoclonal antibodies or mouse monoclonal antibodies. We have various libraries here, alpaca library and human libraries for phage display, searching for antibody binders. And we have transgenic mouse capabilities to produce fully human antibodies. So any antibody that you need, basically, we can produce it with some of those technologies that we have available in-house. Our most recent development was more targeted toward membrane proteins. So membrane proteins are very hard to produce, so we're coming out with a different strategy, essentially, almost like the way the vaccines, mRNA vaccines work. We'll use mRNA to produce the proteins inside the antigen inside the cell, and that would make it easier especially for membrane proteins to be produced, and can speed up the workflow. So that way we can shorten the workflow to 8 weeks. We can develop antibodies and produce them. This is another excellent instrumentation system. This one is for protein antibody purification. So if you're doing a lot of screening in-house and you're producing a lot of various proteins that are tagged in some way for affinity purification, or you're purifying a lot of antibody clones, then you can use this system to essentially purify 24 samples within 1 hour. So each sample can be up to 50 milliliters or can contain up to 100 milligrams of antibody or protein. You basically put your sample inside the lysate or the secreted cell culture. You can put the complete cell culture inside the instrument and use this magnetic beads to do affinity binding, bind the protein of interest or the antibody of interest and purify it in a very fast and efficient way. It's a very automated system, and you can produce, essentially you can do 96 samples a day with one of those instruments if you have this kind of throughput. Of course, we have also very strong expression capabilities. We have been focusing especially on our TurboCHO platform. We have a proprietary TurboCHO platform in-house. As I mentioned, we can produce, basically, if you give us a sequence, we can produce everything that we can synthesize the gene, we can produce the plasmids, we can produce the antigen, and we can produce or the antibody within 5 days. So very fast, we have different levels. You can see how it maps here. Different levels could be used at different stage of the development process. Okay, so for lead generation optimization, we also have a lot of antibody modifications and purification services. We have SC and V generation. We can do cell line development. We can do research-grade cell lines for assays. We can also do cell lines for production. We can produce either polyclonal cell lines, cell pools, or we can produce monoclonal cell lines for either assay or production. Sorry, this is not working that well. So we also have a lot of mutagenesis libraries. As I mentioned before, we have our proprietary gene titan technology that can help us produce a lot of libraries. Those are usually very useful for at this stage of lead generation, especially the optimizations part. We can do various libraries, saturation scanning. We can have site saturation when we vary the amino acids in a specific site, or we can vary the site itself. We can do combinatorial libraries and so on. So basically, we have great support for lead optimization with these libraries. We can do antibody humanization. We have proprietary machine learning and AI capabilities. We can basically, once the template is selected, we can do a lot of optimization in humanizing the antibody. We can do a lot of screening, depending on the type of assay that you would like to see. And then we can produce certain number of clones, and we can deliver them to you basically after the humanization. For preclinical development, of course, we can support candidate validations with antibody and protein expression. We can develop anti-dual type antibodies, ELISA kits, various assays. We can support IND studies. As I mentioned, we have already done many IND studies with various companies, and we have various preclinical grade services related to that. I'm not going to go through all of that. This is just for our plasmid preclinical grade. A lot of parameters are being analyzed and QC'd to deliver very high quality plasmids for your preclinical studies. As I mentioned, we can do anti-dual type antibodies. We have one of the biggest libraries of off-the-shelf anti-dual type antibodies, but we can also do, of course, anti-dual type antibody service, where we would develop the anti-dual type antibodies, and also the complete assays for you, for your studies. We have over 40 different assays that we can support, a lot of CDC, ADCC, neutralization assays, growth inhibition, other assays that we can support during development, preclinical or clinical development. And then here is a little bit more on the biologic CMC, depending on the target you have. Is it naked antibody or ADC? Is it bispecific antibody or protein? We have some here timelines that you can look at, depending on what you're developing and actually to what stage you would like us to take these. Those are some approximate timelines. We have done already many projects. We have done over 18 IND approvals through different regulatory agencies, and we are a one-stop solution. Essentially, we can support for IND filing. These are different manufacturing capabilities for your preclinical studies. We can produce basically CITES fluid. We can develop hybridoma. We can develop recombinant antibodies. You can see some indication here of the scales that we can do this up to and the quantities we can produce. For clinical development, this usually goes to our CDMO division, ProBio. As I mentioned, we can do cell line development, cell line banking. We can do manufacturing. We can support clinical trials or GMP manufacturing, and we can do a lot of the analytics that are required for that. This is some timeline for preparing cell banks and cell bank storage, basically various assays. During our GMP manufacturing, we have basically single line of production that doesn't have any cross with any other production lines. We do comprehensive technology transfer. We have single-use systems. We have zero crossover production lines, and we have cGMP quality systems. Sorry, it's not. Oh. It's not reacting the way it's supposed to. It's not moving. Oh. Okay, so just very quickly on the partnering with GenScript. It's not moving. So basically, we work with many companies. We can partner on different stages of the project, anything from research to IND filings to GMP to clinical trials and GMP production. We have dedicated project manager that would be assigned to you. We can prepare timelines with the sub-projects. We can keep you informed during the whole development. We have all the quality systems required for preclinical and clinical work, and basically the production capabilities at scale. And again, I will be happy to take your questions. Also, we are at booth 425, and we have some posters, AL 53 and 54. Feel free to stop by if you would like to talk to us after the presentation. Thank you. They're tired.
I mean, your slides were clear, and
Okay.
everybody, I think, will come.
Okay, thank you. Thank you for your attendance. And again, feel free to stop by our booth if you like. We have a lot of interesting services and instrumentation. Feel free to take a look. Thank you.
Exactly. Thank you very much. Okay.